Hello, good morning. Thank you so much for having me here. One question, are you sure that you want to listen about molecular mechanism in this Puerto Rico morning? Sunny Puerto Rico morning? Okay, I'm very grateful for you to inviting me to give this talk. And I think we are going to talk about the same thing in the whole morning, but from a different angles. And I think that's going to be very, very interesting. So, this is where I'm coming from. I work in two different institutions, an academic and also a private clinical trial center. So I will give you my overview from both places. And my cardiovascular mentors. So I consider myself an IPC product, and all the way from the IPC founder, Dr. Alan Mentor, who introduced me to my CV mentors, and it's everything I will present here. You know that you all will be on the shoulder of giants, so I'm very grateful to them. And also very grateful to my teams that all the work that I'm usually the one presenting here, but they do a lot of work, so they deserve all the merit that I'm going to share with you. Okay, the first time you hear about immune metabolism, you tend to think, okay, this is simple, that you have obesity and these patients tend to be inflamed, right? It's more complex than that. I have a colleague in my institution, he's a cardiologist, and he has a whole line of research on immune metabolism. And he told me, no, Álvaro, it's not about that. This is about the metabolism, the intracellular metabolism of the immune cells. So going inside the cell, the immune cells that are in the skin, and looking at their metabolism. And depending on the metabolism of that cell could be more pro-inflammatory or anti-inflammatory. So this is quite complex. It's all the way back to 1884, when it was noted that some patients with meningitis, they exhibit transient diabetic syndrome. Sometimes they even didn't know that this patient have meningitis because of the diabetes. And since then, it has lots of publication on this topic. First, starting with macrophages, findings about macrophages in the adipose tissue, then moving to the concept in the 70s about insulin resistance, then our TNF that changed our lives in psoriasis, moving on to Janus kinases, and then demonstrating the role of macrophages into adipose tissues and insulin resistance. If we go to the definition, this is complex, but I like the word bidirectionality, because I think that this bidirectionality is driving us crazy in psoriasis. Because if you hear my previous speaker, we don't know what's first, the inflammation or these metabolic abnormalities. We know, my father was always telling me that logic is not always reality. So logic is telling us, okay, you have the inflammation in psoriasis and that moves into other comorbidities, but could not be the case. But I want to be provocative during my talk. If that's not the case, and we are only treating cytokines, we are inhibiting cytokines, can we cure, for example, psoriasis only by treating cytokines? So during my talk, I will challenge this. But this bidirectionality, this, let's say, interaction and changes that drives immune activation to metabolic changes and vice versa, is what I will cover here. And I want to talk about this from different organizational levels. So the main thing is going to be intracellular. So it's called the metabolic reprogramming, gene expression. But also never forget about the interaction between cells, cytokines. And also we can talk about the interaction with the environment, which is diet and gut microbiome. So this is the concept that I like to talk about, because when we talk about obesity and inflammation, obesity is at the root of psoriasis, and everything starts at the adipocyte. If you can see there, obesity can trigger inflammation, and this is by activating intracellular hypoxia, lipotoxicity, ER stress, and this is attracting chemokines. The adipocytes are secreting chemokines that are inviting macrophages to the party of inflammation. And also another immune cells that then secretes cytokines, and this produce insulin resistance. So this is a cycle, but if you see, obesity is at the beginning. And I like this paper published in Nature, which is the first one that has a huge overview of this immune metabolic topic. And now I like to talk from flies to humans. Why? Because at the beginning, from an evolutionary point of view, if you go to the Drosophila melanogaster, it has only a fat body. And this fat body was serving to store nutrients, to defend against pathogens. And then through evolution towards the mammals, this differentiate into 3 different compartments, which is adipose tissue, liver, and immune system. But there are some interconnection. There are some conservations in the pathways, some crosstalk among them. And if you see the pathway, the main 3 is TNF, which we know, insulin, and the TLR. So this is suggesting that these diseases can go together, and these diseases emerge in clusters. You can put there not only obesity, diabetes, and cardiovascular disease, but also psoriasis. And this cluster tends to go to aging and premature death that we should worry about. So this is an example of what's happening intracellular. And this is more complex because it's not only intracellular. We need to look at the receptor level. We need to look at the kinase pathway and also to the gene expression. And interestingly, when we go to the intercellular communication, we now know that based on these evolutionary changes, we now understand that when we have fluctuation in nutrients, some hormones can behave as cytokines, and some cytokines can behave as hormones. So now we can understand why when we have fluctuation in nutrients, such as obesity, for example, or what's happening in our society, we can have this inflammatory epidemic. Okay, when you start a paper, when you write a paper in psoriasis, which is the first sentence usually? Psoriasis is a chronic inflammatory disease. After my talk, I would love to change that. And maybe we change to another concept. Psoriasis is a metabolic inflammatory disease. Let's see. And I like to focus on this paper published in the Journal of Investigative Dermatology. It's talk about spatial proteomics. So we've been hearing a lot about single RNA sequencing, about spatial transcriptomics, but this is just giving us information about transcriptome abundance. But this, at its best, is only promising that it will have something to do with the biological function. There is still proteins, and sometimes there are some, for example, traduction regulation that can change this. So we need to go also to the proteomic. We still don't know well the spatial, the architecture, the machinery that is happening within the skin. What's happening there is the question of this paper. So what did they do? They took biopsies from psoriatic lesions, other biopsies from healthy controls. They did laser capture microdissection, and they divided the skin into 4 layers. Outer epidermis, inner epidermis, dermis, and subcutis. They did mass spectrometry. So they found 6,000 proteins in there, and in the different layers, the more important was the inner epidermis. And they found 1,600 differentiated proteins between the inner epidermis in lesional and non-lesional. So I want to show you the results of this paper, not within the paper, but within this commentary from Dr. Ward. And here you have a sentence, look at there. I'm going to put you here. Scientists demonstrate psoriasis as a vertically stratified, metabolically reprogrammed inflammatory condition. This is tough, right? Difficult. At the beginning, I didn't understand this. What's vertically stratified? So I'm going to show you this. Remember, vertically stratified, metabolically reprogrammed inflammatory condition. So here you have the idea. Outer epidermis, what you find there, so the idea of vertically stratified is that you can see that when you divide into different layers, what you find is different. So outer epidermis, some proteins there, more complex than expected. Then subcutis, nothing significant. Dermis, IL-17 pathways, neutrophil, chemotaxis, and then within the inner epidermis is where everything is there. We have innate immune activation, but more interestingly, the whole cholesterol pathways is overactive. And here you can see, and you can see in that orange color, is that the whole pathway is not only activated, but the precursor of of the target of statin is very, very increased. As you can see there, the proteins related, the precursor of the target of a statin is really increased there, but the whole pathway of cholesterol is overactive. And they conclude in the paper that this redefined the psoriasis, the disease, as a metabolic reprogrammed inflammatory disorder, potential new targets. Maybe there you can explain why there is so much noise about statin and the role of statin in psoriasis, some meta-analysis showing mild results, but maybe it's just if we change the target to the previous enzyme, we could find something. They of course, think about GLP-1 inhibitor, and also about targeting topically or spatially targeting lipids in the skin. So I know someone in this room who could be interested in commenting later. And let's move to other papers. So, psoriasis immune metabolism, I promised you that this was at the intracellular level, and you can see here the kind of studies. Basically, not all the cells are altered in the same way. If you differentiate T cells, they have a more lipid-rich metabolism. If you go to keratinocytes, more glycolytic. In both, the mTOR, the target of rapamycin, is activated, and also they always mention gut microbiota here. These are some potential targets that I'm not going to get into the detail here, just to think about these enzymes, about statins. Also, they talk about IL-17 pathways that we will cover later, GLP-1 inhibitors that will be covered later during today's meeting, so I will not stop there. And this other study I like is spatial transcriptomic, because it's not only about differentiate the metabolism of T cells or of keratinocyte, but also the subset of T cells. So what they find here is that every different T cells have its own impairment and its own potential target. It's not the same Tregs, it's not the same CD4, it's not the same CD8. So we are just at the beginning of discovering the root of our disease at the local level, at the skin. So I went to other fields where they have been advancing this more. For example, cancer. And you can see there that in cancer, they have already successful targets in immune metabolism, for example, the mTOR signaling, the glutamine metabolism, or other, as you can see in the slide, and I won't get into the detail, just having that there so you can see. Okay, so I went through this molecular mechanism. How does this do into the connection with the environment? Some ideas from our lab. So remember, everything related to obesity inflammation start in the adipocyte. And maybe this is the reason why we still don't understand the directionality, maybe obesity driving psoriasis. Maybe this is the reason why we still don't know the directionality if it's from psoriasis to cardiovascular disease or vice versa. And this is, for example, another paper from our lab where we found that when we have our, let's say, biologics, we are treating this symptomatic with the cytokines, the product of the T cells, we have this complex response, but when we look at the inflammation, even when we have a patient, for example, like this, with a high BMI, we treat the patient, he's like that, but he still has inflammation. So we still have some work in these patients, and that, I think, is the main reason why we are here in this symposium today. Also, we need biomarker for immune metabolism. So we proposed this in a recent paper where we have monocyte, which is an inflammatory marker, and HDL, which is a metabolic marker. So immune metabolism also can be translated easily into clinical practice. And I was talking about gut microbiome, which again, in other specialties, they are doing fecal transplantation, and we found a signature. This is already accepted in the British Journal of Dermatology, and we found a microbial signature which can detect which patients are going to have atherosclerosis progression. So this axis between skin, gut, and the heart is something that we also consider in immune metabolism. I'm not going to talk more about statins, which for sure we need to consider, but I'm going to talk about the Mediterranean diet, because we have published this paper, and I think this is an example of this immune metabolism, because our patients improved 4 points in the PASI score without losing weight. So it's not only about the weight by itself, but the quality of the food that we give that can improve the metabolism and the immune metabolism of our patients. So again, advocating for a holistic approach in our patients. And then moving to cardiology. So this is the kind of images that I receive when we do our research in Madrid. And I went to my cardiologist and he said this to me, Álvaro, this is my whole day. Immune metabolism in atherosclerosis, a new understanding of an old disease. So they are all the way moving now to inflammation, and this is the interesting part. Do you remember those papers saying, are Th17 cells good or bad? I remember that. Are macrophages good or bad? It depends on the metabolism. So what they have discovered is that depending on the metabolism of the cell, they can go to be pro-inflammatory or anti-inflammatory. So again, they are looking for targets, enzymes, or pathways in order to improve the disease at the root of it. This is a game-changing paper in atherosclerosis, published in Nature. Imidazole propionate, a metabolite, has changed everything. It's a very complex paper from a team from Spain, and it's changed the way we see the disease, atherosclerosis, from cholesterol at the beginning, then inflammation, and now metabolism. So it looks like cancer, oncology, and then we, dermatology, we need to build on this field in order to improve our disease. Even the arteries are inflamed, and the metabolism there, we can now measure and improve. So I will just finalize by provoking the room. We want to cure psoriasis. Our drugs are being symptomatic, only treating the cytokines, and we are maybe silencing it. But can we cure psoriasis if this is a metabolic disease, if we are not doing anything on this? So maybe we can discuss on this and see how we can design the next trial, including this, in order to build new standards for our patients. And with this, thank you so much for the attention.
Well, thank you. I think we have time for one or two questions, quick ones. Andy?
So you didn't mention in the immunometabolic pathways IL-23. So IL-23 we know is important, right, in psoriasis pathogenesis, and we always talk about it as upstream. But do you think there's upstream of IL-23, an immunometabolic dysregulation that's increasing 23 that then, so where do you think 23 fits into everything you just showed?
Yeah, so I expected some questions. So I think that one issue is that this field has been building chronologically. So TNF is a lot of research, then IL-17. So there is no research on IL-23 on immune metabolism and how interaction and all the interactions. So I think that is still to be built, and for sure is part of the whole process. I think that's my idea.
Okay, well, thank you. I think one final one, quick one.
Thank you, Álvaro. That was wonderful. Looking at the skin as an organ, of course, cholesterol metabolism is very important. We have lamellar bodies, with the skin integrity is functioning on lipids. How specific to psoriasis would this problem be? And is there like a chicken and egg thing? When I hear your talk, I think about we treat porokeratosis of the skin with cholesterol creams. Yeah. So there is definitely something going on with lipid metabolism. So is it consequence of damage, or so what comes first? And shouldn't we look at all skin diseases in terms of lipid metabolism?
Yeah, so at least in psoriasis, and there have been some trials in other diseases such as acne, atopic dermatitis, of intervention with local statins and these kind of things. So they are at the root of psoriasis for sure. And it has been tested in many different studies in knockout mice with cholesterol genes, and showing how they deplete the inflammation in the skin. So for sure it's at the root of the disease. And that is the main question, because we now, even with our best drugs, if we stop the drugs, most of our patients relapse, even in every study that you do with the best. So there is still something there at the root that we need to go. So maybe it's this or maybe other parts, but this is at least part, and yeah, maybe in the next talk we also hear about that, I think. Yeah.
Well, thank you.